I had meant to do a couple of things this morning before starting out. Actually, I did a bunch of stuff this morning, but not actually that has to do with what we're doing here. One, my son's now registered for actual health insurance. Yay. Got his social security number yesterday. It felt very weird.
He's a person.
Yeah, he's a person now. But then also, I've had a number of students who have sent on to me errors they found in mostly the syllabus online, but those kinds of things. So I'm trying to catch up with those extra credit submissions. The problem with those is if there's an error that somebody's found, like if you've looked through your syllabus and you found an error, it's actually in every copy of the syllabus for all 6 classes that I have online. So I have to go through and make the changes in all 6 classes. And I was going through one set that somebody sent me that they found 10 different errors. So I had to make 60 changes. And Blackboard is not really the easiest system to make those kind of changes in. So that kind of chewed up all my morning. While I was doing it, I found a couple of errors on my own that nobody had pointed out yet. So I fixed those too. What I had planned to do, one of the things I planned to do was to fix this figure. So last time I was talking about how there's an error in this figure, which is not, oh, not this figure, sorry, that's why this looks funny. This figure here. Which was, I pointed out that this might look strange that there's a solid line here going to the adrenal gland while everything else has a dotted line going to it. That actually is okay, that's correct. The error is that there's not supposed to be a synapse there. That solid line is just the axon that's coming out of whatever location here. So what I was going to do was I was actually going to go into Photoshop, kind of thing that I have on my computer, and erase that synapse and just make a continuous line there so that it'd be correct. And then send that corrected image to the publishers and say, that's wrong, gotta fix it. Never got around doing that this morning. The other thing I was gonna do was ask the department chair, who's in the office next to me, if anybody has put in a work order about getting this projector fixed. I've heard several people complain about it. I just don't know if a work order was ever put in. But I think all that needs to be done is that a new lamp needs to be put in, and it's just that done. So some of these lighter elements on the screen, like the dotted lines or the very thin lines, are harder to see, or gray text on the web page that for the book or other things. So I think they just need to change the lamp, and I don't know when that's going to happen. I think actually we might have put a work order in for it at the end of last semester, and they just never did it. But until then, we'll have to deal with in this room that the projector isn't perfect. I am recording what's on the screen, and the screen is not recording from the projector, it's recording from the computer. So if you have trouble seeing stuff here in class up on the screen, if you go back to the recordings, you'll be able to see things just perfectly clearly because it's fine. So I just want to apologize sort of universally for that issue until it gets fixed, which hopefully will be soon. Yes?
The recordings of the,
yeah.
Are there recordings of the actual lecture?
So this fancy microphone I got right here is picking up my voice. I used to do it with my phone, and the phone microphone just isn't enough, and I'd walk over there and you couldn't hear what I was saying. So I got the fancy thing. And also I discovered that I can record lectures. Google has this thing called Hangouts, which is sort of a video chat feature. And within Hangouts, there's an option to do what's called a Hangout on Air, which is a video chat that ends up getting saved as a YouTube video, so it can be broadcast. And so I do that. So if you were watching the screen while I was setting things up, I was doing that. And it captures whatever's on the screen as along with the audio that goes with it. In the syllabus, after where the course schedule calendar is, there's a section that says online resources here. And this very minor seeming sentence in the middle of it says this is a link to the YouTube channel. So if you click here, you'll go to, actually, since I'm logged in as me, you won't see this. Instead, you'll go to something like this page here. And you can watch the video here. So, well, you probably can't see it because of the projector, but there's this little box under here that says live now, because this recording right now, people could watch in real time streaming, which I wasn't aware that's how it worked exactly, but apparently new functionality to it. But when I'm done here and I just hit the stop broadcast button or whatever it's called, then this video will, well, since it's streaming now, I don't know if it has to be uploaded or not, but what used to happen is when I finished recording something, I'd hit stop, and then about a half hour later, it would be available here on YouTube, because it has to be processed by YouTube to be a watchable video. So you can go to these sorts of things. Now, my plan is with this course, which is the traditional lecture style course, to try to capture all of the lectures. I didn't capture lab this week for either group, and I really should have, but I did it, I think, last semester for my MP2 class. But so I want to capture a lecture here, which of course all of you can use if you need to go back and watch stuff again, or if for some reason you missed class and you want to see what you missed. But also so that my other classes that don't have the traditional lecture format, I don't spend as much time with them face to face, they can kind of go in and watch those videos along with the other stuff that I have online. But this resource is available. As I was explaining where that is underneath these calendars, I remembered I needed to point out to you, there are 2 course schedules in the syllabus, and this has to do with the fact that I'm using one Blackboard shell for the whole course. So one schedule is based on the Monday lab, and the other schedule is based on the Wednesday lab. If you're trying to figure out which is which, it's just a matter of looking at the date for the lab meeting. So this is 125, which was first Monday, 2-1, which was this past Monday. So this is obviously the Monday schedule. Along with the fact that for the 16th, it tells you that there's a special Tuesday meeting for lab, because that's the day after President's Day, and we follow Monday's schedule that day. And then the one after this would be Wednesday. Also, just remember, because I'm kind of, I don't want to say obsessive, because it's not like I actually have a psychiatric disorder, but it just, I don't feel entirely comfortable unless I put the Monday one first and then the Wednesday one. Okay. So Monday's the first one, Wednesday's the second one. That's just the way that that makes sense in my mind. So that should be hopefully self-evident. But yeah, so if you're in the Monday lab group, pay attention to these dates. If you're in the Wednesday lab group, pay attention to these dates here. And that's just a quirk of the fact that you're all in the same Blackboard shell course. Like I was talking about, lab quiz due dates are a little weird in here for you guys. Okay, anyways, back to actual content. So I finished up last time by talking about this figure here, which I spent a lot of time talking about this little funny thing that the adrenal gland has a preganglionic fiber going to it. Aside from the error that I'm talking about, that there should not be a synapse represented here, where that falls on the picture is kind of unusual, maybe. The reference figure that I gave the art department to develop this figure from, I think had all of these organs in a column like this, so they sort of follow the same format. And it didn't occur to me, and I'm not even sure it would be a great idea, that the adrenal gland maybe should be over here in line with these ganglia. And I'll explain why that is a little bit. But just to rehash real quick what I was talking about, the dots here represent neuron cell bodies located in the spinal cord. For the sympathetic division, it's the thoracic, meaning T1 through T12, and upper lumbar spinal cord, meaning L1 to roughly L2, or the border between L2 and L3. So that's where the neurons are. In lab, I talked about where they are in relation to a cross-section of the spinal cord. Those axons come out into the spinal nerve. They very quickly leave the spinal nerve and enter into the sympathetic chain ganglia system here, where they're going to make synapses. There are some suggestions here drawn in of synapses, but the only actual synapses represented are up here. That's not to say that this is the only level that real synapses exist. There's synapses everywhere. But just to try to keep this figure less cluttered, up here where there's nothing else to deal with, they have the target ganglion neurons in the drawing here. And then you see their dotted lines coming out. And these happen to be going to the heart and larynx in the drawing, just because that's kind of the area that they'd be heading for, the upper thoracic cavity. There are some ganglia that are superior to this T1 level. They're called cervical ganglia. And anything that synapses on these ganglia are coming from the T1 through upper lumbar, the thoracic through upper lumbar region. But these ganglia are headed to more superior locations to control what's going on there. Mostly head and neck targets, like the iris of the eye, the glands for salivation, mucous membranes, and that sort of thing, are most of those targets. But they're getting their input originally out of the same sympathetic region of the spinal cord. And then down here where I said they don't actually show anything coming out of the chain ganglia, any dotted lines, that's because what they're showing is that besides synapsing on the chain ganglia, some of these preganglionic fibers can also continue on to other ganglia. So this celiac ganglia and the superior and inferior mesenteric ganglia are referred to collectively as the prevertebral ganglia. There's another name I can't remember offhand, but it's in the textbook. And that name, prevertebral, just means that it's anterior to the vertebral column. So it's sitting sort of in this area. Superior and inferior mesenteric is meaning that they're like next to the mesentery, or basically the intestines. And then the celiac ganglion is up here. Sometimes the celiac ganglion is also referred to as the solar plexus. Okay. I think solar, obviously the word solar as in solar system is referring to the sun. I think the meaning of that word is more based on pre-medicine understanding of the body, that was sort of like the center of the body, and everything was radiating out from that, like the sun in our solar system. But it's just a plexus or a network of nerves where there are synapses between preganglionic and postganglionic fibers. And a lot of the fibers that emerge from that ganglion go out to control a lot of the major organ systems. So again, in this drawing, pretend there's not a synapse right there, that one of these preganglionic fibers continues on through the celiac ganglion and goes directly to the adrenal gland. Sorry, you had a question.
The prevertebral ganglia are 3, which include, you said the
So this is the celiac ganglion, which I said also sometimes called the solar plexus. And then these two are the mesenteric ganglia, the superior mesenteric and the inferior mesenteric. The word mesentery refers to the membrane that connects the digestive organs in the abdominal cavity to the wall of the abdominal cavity. So as a term, it kind of just means guts, as far as where those ganglia are located. Preganglionic fibers synapse in ganglia. Okay. For the sympathetic system, that's actually a fairly complex system or network. There are a lot of branches. One neuron right here could send a branch off of its fiber up somewhere in the chain, or down somewhere in the chain, or out to one of the prevertebral ganglia, or to the adrenal gland. Okay. Lots of possible targets there. And I said up in the chain and down the chain, but also could synapse right at the level of the chain where it emerges from the spinal cord. So there are branches all over the place. I think on average there's a couple of dozen branches for each preganglionic fiber in the sympathetic system. So there's a lot of branching. And that branching means that all of these things get activated at the same time. Okay? The same way that this light switch here has one wire coming out of it, and then it branches to go to this fixture, and that 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 fixture. So there are 12 branches off of that one wire to activate all those fixtures. So there's probably 24 branches off of this fiber here going to all sorts of different targets. So when this cell is active, all of those targets get turned on. So blood vessels divert blood from the digestive system to the skeletal muscles. Sweat glands become active. Pupils can dilate. Adrenaline is released and goes throughout your body. Your heart rate increases. Your bronchioles and bronchi, the tubes in your lungs, will expand so that more air can get in. Your breathing rate might go up, those kinds of things. All of that happens at once because of all of the branching in the sympathetic system. Speaking of sweat glands, I just want to point out in this picture, again, to keep the picture from getting too complicated, they have one fiber coming out from the chain ganglia going to a little drawing of a patch of skin. Okay? That's not to say that there's only one connection out to the skin. Okay? Your skin's all over your body, so it's got to get connections from everywhere. So this is going to skin, let's see, what is that, T8? So that's going to be, a patch of skin maybe right about here. A fiber coming off of the L5 ganglion, L5 level chain ganglion, would be going down to, I think L5 is like the top of the foot or something like that. I can't remember the map of those nerves very well offhand. But so every piece of skin gets input to its sweat glands through fibers, and they're coming out of the ganglia that correspond to that particular region of the body. Okay, it's not just everything comes out of here. Just for the sake of this drawing, they only put one little patch of skin in the picture, but the sweat glands are controlled by sympathetic output. Okay. The other picture here. Did I even put this picture up on the screen at all? Okay. I will often double check things like that a lot because on Tuesday I teach 3 A and P2 classes in a row. I teach you guys 11, I teach another one at 1, and I teach another one at 5:30. And I'll often get confused about what I showed you versus what I showed those other 2 classes on the same day. But they're the 1 a week classes, so I have to sort of cover everything for them. So for you guys, I just started out, I did like 10 minutes worth of information. And then I started all over with the next class, and then I did the whole thing with them, and then I started over again for the next class and did the whole thing with them. So by that time, I've forgotten where I exactly stopped with you guys. So I'm going to often ask, did I show you this? Because I would have showed it to my other classes. Anyways, so this is a very similar picture, and it's sort of set up, except instead of showing you the output for the sympathetic side, what's coming out of the thoracic and upper lumbar spinal cord, it's showing you the parasympathetic output. So up top, we have the brainstem, and at the bottom, it's referring to the sacral region. There is actually a little kind of glitch in this picture too, which I'll talk about when I get down there. But they have left in the preganglionic sympathetic connections. They just don't show the postganglionic stuff. Personally, I would have rather that they left everything blank, but the artist, for whatever reason, decided not to erase everything. So, and also they left in the connection to the skin to control sweat glands. Again, this is part of the sympathetic thing. So it'd be better if it wasn't in this picture, but for whatever reason, they didn't erase it. But they have added in what's new is the parasympathetic output. Now, from the brainstem, parasympathetic preganglionic fibers are going to be traveling within cranial nerves. Okay? The superiormost nerve that's involved is cranial nerve number 3. Does anybody remember what cranial nerve number 3 is called? That's fine.
Your eye movement.
Sorry?
Ocular.
Yeah, oculomotor.
Thank you.
For eye movement. So largely what that nerve is responsible for is going to the extraocular muscles and making them contract and move your eyes. But there's also a small subset of fibers in that nerve which go from the brainstem to the ciliary ganglion, and then the ciliary ganglion sends fibers to the iris of the eye. Okay. The iris of the eye contains smooth muscles in it. Actually, 2 sets of smooth muscles. One set is radial fibers that go from sort of as center as it can be out and radiating out from the center. Of course, the center is the pupil, the hole, but from the edge of the pupil out, radiating. And then there are circular fibers, which are a sphincter or an annulus of fibers that surround the outside of the iris. The fibers coming from the brainstem here, they come from what's called the Edinger-Westphal nucleus, which this is a typo. There's only one D in Edinger. And I thought I told them that, but maybe they didn't fix it in the picture. So from that nucleus, fibers go to the ganglion, and then from the ganglion they go to the iris, and those control the circular fibers. When circular fibers contract, any kind of sphincter type muscle, the thing closes down. So for the parasympathetic system, it causes pupillary constriction, which is a rest type of response. It keeps light from hitting the retina so that photoreceptors can regenerate from being bleached and that kind of thing. Right now, right now, standing on the rather dark side of the room, my pupils are fairly constricted. Nope, that's not right. Said it wrong. On the dark side of the room, my pupils are fairly dilated because there's not a lot of light getting to my eyes, and so the pupils dilate. But as soon as I step in front of this bright light from the projector, they constrict. Okay, so from dim light to bright light, information ends up going to the Edinger-Westphal nucleus saying that the brightness of light hitting the retina has changed. And so it sends out a signal through the ciliary ganglion to cause the pupil to constrict. Okay. Just a reflex, bright light, straight to the pupils. Okay. And then when I step out of the bright light and now the light levels have dropped, that signal ends up getting to the thoracic spinal cord which sends a signal through the chain ganglia to the radial fibers, causing them to contract, which dilates the pupil. Okay, so constricted because of parasympathetic input, dilated because of sympathetic input, based on the change in light levels. Wasn't really planning on talking about that specific example right now, but there you go. So that's in cranial nerve number 3. Okay. Then the next 2 nerves are cranial nerves 7 and 9. What are those 2 nerves? What are they called? 7 and 9. Say it again. The facial is number 7, and 9 is glossopharyngeal. They both are responsible for transmitting taste information from the tongue, that sensory information. Tied in with that, they're also responsible for controlling salivary glands. So the parotid gland, the sublingual gland, and the submaxillary gland, those are the salivary glands. And based on taste information or smell or sight or just thinking about food, the parasympathetic system will send signals to those glands through, there are 2 ganglia that are part of that circuit that will cause the salivary glands to produce saliva. Getting you ready to be able to start digesting food. The last cranial nerve that's involved here is number 10. What is cranial nerve number 10? Vagus. Right? Vagus nerve is only about autonomic function, as far as we're concerned. Technically, there's like 1% of it that does something else, but we don't ever talk about that in this course. So the vagus nerve is an autonomic fiber nerve. And more specifically, it is the rest of the parasympathetic preganglionic fibers that are going to targets that are not in the head and neck. Okay, so 3, 7, and 9 go to the head and neck. 10 goes to, usually it's described as everything from the heart to the intestines, okay? Which is anything in the thoracic cavity and most things in the abdominal cavity up to or down to the intestines. Again, all of that's preganglionic fibers. And then from the sacrospinal cord, those preganglionic parasympathetic fibers are headed for everything from the intestines down in the abdominal cavity, and then everything in the pelvic cavity, which will be urinary and reproductive structures, which of course you can't see because of the projector, but that's what's down here at the bottom of the picture. Now, I said there's a little snafu with this picture. Where they show the sacral preganglionic fibers originating is off a little bit. It should be coming out of the spinal cord, but they just put the dot in the wrong spot. Every time I teach, I find more and more errors, and I've been talking to the editors, and I was going to send them all of these corrected figures, and then Benjamin was born, and I said, I'll get to you in a few weeks. But yeah, just bear with me. It's a first edition textbook, and all first edition textbooks have errors like this. So anyways, it would actually be coming out of the sacrospinal cord, but those go to the pelvic and lower abdominal organs. Now, what's impossible to really see in this picture the way it's projected on the board, and not so obvious when you're looking at it directly on the screen, are the postganglionic fibers. And the reason for that is parasympathetic ganglia, which are collectively called terminal ganglia, are located near or even in the target organ. So up here in the head and neck, we can see the terminal ganglia. The ciliary ganglion, which happens to be located in the orbit just posterior to the eye. The, I can never pronounce this correctly, pterygopalatine ganglion, which goes to the lacrimal gland and mucous membranes. And then the otic ganglion, and this one isn't labeled, I can't remember what it's called, but those ganglia there are near the target organs, but we can see them easily as being physically separate from the organs. But then when we get down to the thoracic and abdominopelvic cavities, for the most part, the ganglion is actually just nervous tissue incorporated into the wall of the actual organ. The best example of that is in the digestive system. There's a submucosal plexus and a mesenteric plexus of nervous tissue in the wall of the intestines and the stomach and all that stuff. That is essentially the terminal ganglion for controlling the digestive organs. It just incorporated into the wall of the organ. It's not a separate ganglion, it's just there. Actually, all of that in the digestive system is often referred to as the enteric nervous system. I don't know if this is entirely accurate. I don't remember where I read it, and I don't know if I ever actually verified it. But if I remember, I read somewhere that there is as much nervous tissue in the digestive organs as there is in the brain. Okay. If you think about the intestines, they're, I can't remember, let's say 33 feet long. I think that's a little overly long, but really freaking long tubes. Okay. And all along that tube, there's a good bit of nervous tissue. Okay. If you took all that nervous tissue and balled it all up into something that's about the size of your brain, it'd be, well, about the size of your brain. So the nervous tissue is probably pretty equal in its mass. It's just distributed over a much larger area down in the enteric nervous system. But it's a pretty significant part of the nervous system. And that is pretty much all of the terminal ganglia of parasympathetic system that controls the digestive functions. We will talk about digestion, of course, much more in the digestive system chapter, and we'll learn that sometimes it doesn't need the autonomic system input. It can work on its own, which is a little unusual for the nervous system, but the enteric nervous system can do that. So the lines that we see drawn in here are solid, and they are the preganglionic fibers coming from the spinal cord or the sacral, brainstem or the sacral spinal cord. Where you actually can see the postganglionic fibers, those are supposed to be dotted lines like in the other picture, but they didn't do it here. But really that would only be these that you can really see that well. From the heart down in this picture, the postganglionic fiber, as it's drawn in here, is probably about the length of a single dash in the dash line anyways, so you wouldn't really be able to tell. And that might be what the art editor decided on. Well, we can't tell that there's a dash down here, so let's not make any of them dash. Not a great idea, but very, very short postganglionic fibers in the parasympathetic system. So that's the layout, the anatomy of the system for the two divisions. Okay, two separate pictures. There is a little overlap in what they're showing, like the sweat glands in both pictures. But remember, the sweat glands are really only controlled by the sympathetic system, which is why they're on this side of the picture for the parasympathetic side. All of the parasympathetic input is going over there. Oh, sorry, this is not parasympathetic. Okay, let's pretend I switched over to the parasympathetic picture. All of the parasympathetic stuff is on one side in the picture. It's not the way it is in the body. You know, you have bilateral structures. You have two eyes, two sets of glands in your head, that sort of stuff. They've just drawn one into the picture. Yeah.
I'm kind of confused. So with those gray lines, those lines.
Okay, so these lines.
Yeah.
Okay.
It's like that you are carrying information from the spinal cord to the digestive system and the.
So to all of the target organs. Yeah. Well, all right, let me modify that. These, the preganglionic fibers, are synapsing in the ganglia. Okay. Then from the ganglion to the target organ, there's a second neuron, a second fiber. information originates out of this neuron. It sends an action potential along this fiber, which goes all over the place because of all the branches. And then it synapses on a ganglion, this one or any of these, et cetera, et cetera. Its target cell, the ganglionic neuron, will then generate a new action potential that goes down its fiber that goes to the target organ.
So it doesn't use like, the parasympathetic, it uses the cranial nerves and here it just goes to the ganglia and then transfer back to the target.
Let me try putting it this way that might clear that idea up a little bit. From the central nervous system, there's a fiber going out, synapsing on a ganglion. And this is true for the whole autonomic system. And then from the ganglion, there's a second fiber that goes to the target organ. Okay. This picture shows us the sympathetic division. Everything originates out of the thoracic and upper lumbar spinal cord. The ganglia are either in this chain here or one of these ganglia here. The other picture is the parasympathetic side. So the neuron that initiates the signal is coming out of the brainstem or the sacral spinal cord. So nerves coming out of the brainstem, and this is universal, are called cranial nerves. Nerves coming out of the spinal cord are called spinal nerves. So in the previous picture, everything coming out of the thoracic and upper lumbar spinal cord, we're traveling through spinal nerves. Here, because we have output from the brainstem, it's traveling through cranial nerves and spinal nerves coming out of the sacrum. And that's just an anatomical issue. That cleared up a little bit? Yep. Okay. Other questions? I think what I'm doing next is actually kind of continuing on with that sort of stuff. Okay, I did skip over a picture here. This picture is a graphic representation of that idea I said with branching. This is showing you all the different possible ways that preganglionic sympathetic fibers can influence a ganglion somewhere, whether it goes up or down in the chain, leaves the chain and goes to another ganglion or whatever. And this actually needs a fourth frame showing the adrenal gland. But you can look at that picture yourself. Shoot, I thought there was a picture in here, I guess not, showing sort of a summary of that idea. Okay, so we have in the central nervous system a neuron that's axon goes out and synapses on another neuron in a ganglion, which then goes out and synapses on a target organ. Okay. Now what I've drawn in here is specifically referring to the sympathetic system. Okay. If I do this again for another neuron in the central nervous system, and here I'm talking about the para, its axon goes out and synapses on a neuron in the periphery, which then synapses at the target organ. Okay. For the sympathetic part of this picture, I've drawn the ganglion neuron close to the central nervous system, in the chain or prevertebral ganglia. For the parasympathetic system, I drew the ganglion neuron close to the target organ, a terminal ganglion. Okay. The preganglionic fiber is short for the sympathetic system and long for the parasympathetic system, relatively speaking. And the postganglionic fiber is, relatively speaking again, long for the sympathetic and short for the parasympathetic. Okay. I thought I had a picture that basically showed this in the book, and there needs to be one. Now, where I am going with this, there's a table in the book right here that is titled Autonomic System Signaling Molecules. Okay. Now the term signaling molecule is a broad term that would apply to neurotransmitters or hormones, as far as we're going to use it in this course. And there's a reason why I'm being general about it right here. But preganglionic sympathetic fibers, postganglionic sympathetic fibers, preganglionic parasympathetic fibers, postganglionic parasympathetic fibers. All this is laid out in the table, and it tells you what each one releases. Preganglionic fibers, for either division, release acetylcholine at the synapse they make. Okay. You remember acetylcholine? Where did you first learn about acetylcholine? Yeah, in the muscle system, about the neuromuscular junction. Okay. The neurons that control skeletal muscles, the stuff that you learned about in A&P1, that make neuromuscular junctions with the muscles, they're located in the ventral horn of the gray matter of the spinal cord. Okay. These central nervous system neurons in the sympathetic or parasympathetic system are located in the lateral horn of the spinal cord gray matter, or in a nucleus in the brainstem. Okay, so for the parasympathetic system, sometimes it's from a nucleus in the brainstem, but for the spinal cord, it's called the lateral horn. The lateral horn is right next to the ventral horn. Okay, the cells in the lateral horn and the cells in the ventral horn are actually the same kind of cells, multipolar motor neurons. One way of thinking about it, anatomically, we describe the two areas as the ventral horn and the lateral horn. We could also just say that the motor neuron pool is distributed. The more superior lateral placed neurons control control organs, and the more ventral medial located neurons control skeletal muscles. Okay, it's just one population of cells. They're all very similar. Their eventual targets are different. But when they reach their initial targets, they all release acetylcholine, okay? Whether it's at the neuromuscular junction or one of the ganglionic synapses here or here. Okay. So these cells on a basic level are very, very similar to the motor neurons that you studied in A&P1. Okay. Do you have a question?
So
Looks
the
like
lateral, the ventral controls skeletal muscles.
Yeah, let me make a quick picture of that. Okay, so this is my rough sketch of a cross-section of the spinal cord. So this is the dorsal horn, which is about sensory stuff. And this is the ventral horn, which you learned last time controls motor stuff. This little bump that I put in here represents the lateral horn. Okay. And that's where we find the preganglionic neuron cell bodies for all of the sympathetic division and the sacral part of the parasympathetic division. The cranial part of the the cranial part of the parasympathetic system, that's brainstem. Brainstem doesn't look like this. That's what this is trying to play. So this area right here is about autonomic, and this area right here is about somatic, or controlling skeletal muscles. Okay. We could also just think of all of this area right here as motor neurons. It goes from autonomic to somatic from this position to that position. It's another way of thinking about it. Or I'm using autonomic and somatic. Some sources will say visceral, meaning controlling organs, to skeletal, I guess, to control skeletal muscle. Or I don't know, skeletal, somatic, those sorts of things they would use. But okay, that was a very quick drawing, and it might confuse the hell out of you because it's raw, straight from my brain to the thing here. This picture over here is a little bit more practiced, but okay. So the preganglionic sympathetic fiber and the preganglionic parasympathetic fiber both release acetylcholine, which is actually exactly like any ventral motor neuron. Okay. Anatomically and chemically, all of those neurons are pretty much the same thing. One more thing that's very similar about them, which is the type of receptor that acetylcholine binds to. Okay. I think at the end of AMP1, I talk about this a little bit. If you did not have me for AMP1, this might be new to you. But acetylcholine receptors come in a few different varieties. Who's familiar with that idea?
Sorry.
Acetylcholine receptors come in different types. Remember that from AMP1? Okay. Other people remember that? Other people learn that from their AMP1 professors? Maybe not. Sometimes in AMP1 they don't talk about it. Okay. At the neuromuscular junction, when you learned about the receptor that acetylcholine binds to, whether you were in my class or not, what physically was that receptor? Well, it's a motor system, so I won't be right. But physically, what is it? If you could pick it up and hold it, what would you be holding? No, it's at a synapse, but it's the receptor. Okay, so hopefully somebody immediately went, oh, it's a protein. Receptors are always proteins. Okay. But nobody said that, so I want to cover that. But the actual acetylcholine receptor protein is what type of protein? What happens when acetylcholine binds to the acetylcholine receptor at the neuromuscular junction?
Movement.
Okay, so it causes movement, which means that a muscle's contracting. Okay, why is the muscle contracting?
It's contracting because.
And don't say because acetylcholine bound to its receptor.
No,
I'm trying to get.
because acetylcholine causes the chlor.
Right neighborhood, wrong player.
You talking about the pump?
No. But again, you're in the right neighborhood. Sorry?
Are they?
Okay, you've left the neighborhood.
What's released?
I don't think that's what I mean, but what do you mean by that?
Like calcium release.
Okay, I see what you're saying. So we want to back up from that. But yes, in response to the question I just asked Ali, he said it contracts. And I said, what makes it contract? And so we could go, well, calcium was released and, and all that stuff. Now I want to ask you, why was calcium released?
The voltage of sodium channel.
So you said voltage. Okay. We'll stop you there because that's good. Okay. So there's a voltage change, namely depolarization, that causes calcium release. Okay. What caused that voltage change or that depolarization?
The imbalance of sodium and potassium.
Okay, so membrane voltage is based on sodium and potassium ions location. And by imbalance, what do you mean?
It goes from being negative to positive inside the cell.
Okay, so the inside of the cell is normally negative becomes positive. What makes it positive? Well, directly it's because the sodium moves in. The potassium moving out gets it back to negative afterwards. Okay. How does the sodium get into the cell? Okay, so I'm laughing a little bit because you're jumping back to the beginning of the whole story and you're jumping over the actual thing I want you to say. But Acetylcholine binds to its receptor and sodium enters a cell. Why does sodium enter the cell when acetylcholine binds to its receptor? We've already established that's what we call depolarization. Okay, the concentration drives the sodium into the cell, but how is it getting physically into the cell? Okay, gradient is just another way of saying concentration, but physically, what is the sodium moving through? A channel. What is that channel? It's not a voltage-gated channel because we're starting everything off. This is the first time sodium enters in. So how does sodium first get into that cell? Protein channel. What is that protein channel? Getting really close to putting the two ends together. It is the acetylcholine receptor. Okay. Remember, acetylcholine binds to its receptor. It is a channel that opens up and sodium comes in. Remember that? If the answer to that is no, you need to go back and review some stuff from AP1. But, okay, so that's the acetylcholine receptor that you learned about. Okay. There are other types of acetylcholine receptors. We classify acetylcholine receptors into two types. One is called the nicotinic receptor, and the other is called the muscarinic receptor. Who remembers hearing that? Definitely you guys.
I read it today, yesterday.
Okay, so if you did not have me for A&P1, you probably did not hear that in A&P1. I happen to say it in A&P1 because it's part of explaining neurotransmitters in chapter 12. If you did not have me for A&P1, please go back in the book and look at, all right, yeah, go look at that part of the book. But I realized last semester I decided to revamp that the table about neurotransmitters, and I have a better table for that. But if you've never heard that before, you can go back and look in chapter 12 for the stuff about neurotransmitters. But we're going to cover what we need to here for the most part anyways. Okay. So there are nicotinic receptors and muscarinic receptors. Nicotinic receptors are the ones that you know about. Those are the ion channels that open when acetylcholine binds to them. Okay. And that's what's at this synapse. So acetylcholine binds to the nicotinic receptor here and here, which is another reason why the preganglionic fibers of the autonomic system are just like somatic motor neuron fibers. Okay. They come out of roughly the same part of the spinal cord. They travel through spinal nerves to their target where they release acetylcholine, and then that acetylcholine binds to the nicotinic receptor. Okay, so nicotinic receptor here and here in the autonomic system, nicotinic receptor at the neuromuscular junction. Same thing, causes depolarization of the target. Okay. For muscles, it causes the muscle to contract. Here, when acetylcholine binds to the nicotinic receptor, one of these ganglia, it causes the postganglionic neuron to generate an action potential, which then travels to the target organ. Okay. And the connection at the target organ, what I'm calling a synapse, and some of my colleagues would say it's not really a synapse, but we're going to stick with synapse. There we see another neurotransmitter release binding to a receptor, okay, which is the postganglionic side of things. Now, the postganglionic parasympathetic fiber, okay, this right here releases acetylcholine again. Okay, so same neurotransmitter, different location. What's different, however, is what it binds to, and I'm kind of running out of room here. Okay. On the target organ, acetylcholine is released by the postganglionic parasympathetic fiber, binds to a muscarinic receptor. Okay. Now, a word about words. Okay. Nicotinic obviously sounds a lot like what? Nicotine. Okay. Using the same kind of construction, what would muscarinic be like? Something called muscarine. Okay. These are just chemicals that will activate these receptors just like acetylcholine. The names just come from, in the laboratory, if we want to study these receptors, we can make them do what they're supposed to do by putting that particular drug on them. Okay. Now we can put acetylcholine on the receptor, but acetylcholine is going to activate either type of receptor. We want to just activate nicotinic receptors in the laboratory, we can put nicotine on it, and that'll happen. We want to just activate muscarinic receptors in the laboratory, we can put muscarine on it. Okay. You probably don't know what muscarine is, which is fine. We don't care. As far as you're concerned, it's a chemical that we can use in the laboratory to activate that type of receptor. Okay. But unfortunately, you do know probably what nicotine is. Hopefully not directly. Okay. Right now, don't worry about that. It's just a chemical we can use to activate that type of receptor. A little later on, it'll be relevant why it's actually nicotine that we've heard of. But just two different types of receptor proteins. This one is only activated by nicotine. This one is only activated by muscarine. Okay. Now, the way I like to describe this is using the key and lock metaphor for protein binding. Neurotransmitters bind to the receptor proteins. Okay. A three-dimensional molecule fits into a three-dimensional spot on a receptor, very much like a key fitting into a lock. Okay. And when the correct key goes into the correct lock, you can turn the key and unlock the lock. You're familiar with keys and locks? Okay. So when acetylcholine fits into the physical space of the nicotinic receptor, it causes the nicotinic receptor to open up and allow sodium into the cell. Okay. Just like I could put a key in this lock, turn it, and open this door. What's going on here with the two different receptors is like having two different locks. Okay. And I used to do this, and I used to have physically keys that I could pull out and talk about. So let's pretend I have those keys still. We've changed the lock system in this apartment. But let's say this key here is my classroom door key. I put it in that door, I turn it, it opens the door, we can come in. This key here, let's say that this is the key to the AV room, which is across the hall. And down a little bit. Put that in the AV room door, turn it, unlock it, can go in, get AV equipment. I cannot put the AV room key into the classroom lock and open that. And I cannot put the classroom door key into the AV room lock, unlock that, and open up. You understand how keys and locks work, okay? But I also have a third key. And this is a master key to the department, which means I can put it in any lock in the department and open it. So I can put this key into the classroom door, turn it and open it, or I can put it into the AV room door, turn it and open it. Works in both locks. With me so far? Okay, so the master key is acetylcholine. Activates both receptors, opens both locks. Nicotine is the classroom door key, only opens the classroom door, does not open the AV room door. Muscarine is the AV room door key, only opens the AV room door, does not open the classroom door. Okay, see, very mechanical metaphor for it, but it's a pretty good representation. What's really great about that as a metaphor is that keys have a particular shape, and that's how they fit in and make the lock turn. It's because of the shape of the teeth on the keys. You change one tooth and it will no longer work that lock, or it won't work that lock very well. Okay. The difference between nicotine and muscarine is so great that one will not open the other lock. However, acetylcholine is like both of them. And so they both work that. Very, very simplistic and therefore most likely actually wrong representation of that idea. Acetylcholine is a molecule with sort of two parts, the acetyl part and the choline part. Okay. Nicotine, and I don't know if this is at all close to reality, but it works for an explanation. Nicotine is shaped like the acetyl end, but then looks like something else over here. Okay, so this is nicotine. And Muscarine looks like the choline end, but then has other shape over here. Okay, that's okay. Very crude drawings, very simplistic explanation, most likely in reality not correct, but still explains the idea. So this part of nicotine is the same shape of this part of acetylcholine and therefore will bind to the nicotine receptor. This part of muscarine is like this part of choline, so will therefore bind to the muscarine receptor. Acetylcholine, when it binds to the nicotinic receptor, binds on this side of its molecule. And when it binds to muscarinic, it binds on this side. Very simplistic, probably in reality not correct, but I think it gets the idea across. Like a master key, it can fit in either of the locks. Whereas the individual drugs only fit in one lock and not the other. Everybody follow along on that? Good with that? Okay. So we've left out, or we have not gotten around to the fourth possibility here, which is the preganglionic sympathetic connection. Nope, said that wrong. Postganglionic sympathetic connection. What the sympathetic system releases onto its target organs. What it releases is a molecule called norepinephrine. Okay. Did I talk about norepinephrine or epinephrine at all in class on Tuesday or in lab? Okay. So it's abbreviated NE for norepinephrine. That is spelled, okay. That's not a good place to put it. Norepinephrine. It is a molecule that's very closely related to epinephrine. Okay. And in chemical naming structure, when you have nor on the beginning of something, that means that that's something plus a little extra, a little chemical change. So as far as we're concerned, norepinephrine and epinephrine are very, very similar molecules. Well, as far as anybody's concerned, they're very, very similar molecules. So for now, let me ignore the nor part of the word and just talk about epinephrine. Okay. Epinephrine comes from the Greek. Epi means on top of, and nepher means kidney. Okay. And then INE is a chemical ending. If we said the same thing using Latin roots instead of Greek roots, on top of is ad, and kidney is renal, and then we can put INE on the end of that. Okay. Epinephrine is adrenaline. Adrenaline is epinephrine. They're the two words for the same thing. The reason why there's two words for the same thing is thanks to capitalism. Back in the 60s, there was a company that started selling a drug that they called adrenaline because it was adrenaline. They probably purified it from adrenal glands of, I don't know, cows or pigs or something like that, and sold that. Because they started selling this drug called adrenaline, they made adrenaline a trademark so that other people couldn't infringe on their selling a drug, whatever. It's a trademark. But if a scientist wanted to publish a paper about research on adrenaline in America, because the word adrenaline was owned by this drug company now, the scientist had to get permission to use that word. The way capitalism and trademark law works. Now, if that were the case, and they wanted to publish a paper using the word adrenaline, and they said, okay, can I get permission to use it? The drug company would either say, yeah, feel free to use that word. Here's a letter legally saying that you have the right to use the word that we own. Or they could say, we'd be happy to let you use that word, but you have to pay us money for the right to use it. Or they could say, no, you can't use that word. Okay. So the first option would be the nicest way to deal with it. The second option would be a little bit unfair, basically charging scientists to do the work that they're trying to do. And then the third option would potentially be a way for the drug company to keep scientific evidence out of publication if it maybe said something disparaging about the drug they're trying to make money on. Worst case scenario. So to avoid any of those possibilities, United States scientists decided to stop calling adrenaline in formal writing and switch to the Greek words epinephrine to say the same thing. It means the exact same thing, but the drug company does not own the Greek version of that word, so they don't have to ask for permission. Outside of the United States, scientists publishing papers about adrenaline do not have to worry about the use of that word because the trademark law only applies within the United States. So you will often see both words used to describe that molecule. In common usage in America, where you do not have to ask permission to use a word, we tend to call it adrenaline. But in formal scientific writing in the United States, it's called epinephrine. Any formal scientific writing outside of the United States refers to it as adrenaline. Same thing. Also, what I just told you we call norepinephrine is noradrenaline. Now, for the most part, I probably wouldn't have to belabor this issue too much, but thanks to the internet, I do. If you're looking up information about these chemicals, keep in mind that norepinephrine and noradrenaline, or epinephrine and adrenaline, are the same things. Norepinephrine and epinephrine, or noradrenaline and adrenaline, are not the same thing. But for the sake of this class, they work very, very similarly. And I'll explain why later in a second. So, norepinephrine, formal name for that molecule. Technically, on the internet, if you run into noradrenaline, it's just another name for this name. Now, norepinephrine has its own receptors. Okay? Acetylcholine binds to acetylcholine receptors, so norepinephrine is going to bind to norepinephrine receptors. Technically, we don't call them norepinephrine receptors. We call them, and this is going to just really put a whole wrench in everything I've talked about, adrenergic receptors. Okay, so drug company owns the word adrenaline. They do not own any variation of that word. So scientists can use adrenergic, which is an adjective describing things that are related to these molecules. So they're called adrenergic receptors. The good news about that little wrench I just threw into everything is that we don't tend to use the word adrenergic a whole lot. Okay? There are two types of adrenergic receptors, which are called alpha adrenergic receptors and beta adrenergic receptors. And so mostly we'll just say the alpha and beta receptors. Okay, so you don't really have to use the word adrenergic at all in this course. I understand what it means because I might use it occasionally, but we'll just refer to those as the alpha and beta receptors. Which norepinephrine and epinephrine bind to. Okay. Now, there are two types of acetylcholine receptors, and which type is which is important because they're found in different locations. There are two types of norepinephrine receptors, but for the most part, we're not too bogged down with which type is which. Okay. They are found in different places. I personally do not remember where those places are, which means you personally don't have to worry about it either. There are, I think, 3 types of alpha receptors and 2 types of beta receptors, little variations. The best I know of any actual locations, beta receptors are found on the heart. Okay. Don't get too bogged down with that because it's not crucial for this course. The reason I tend to remember that and the reason why it's worth even mentioning is that there is a class of drug that blocks beta receptors that's used for cardiac disease issues called beta blockers. So obviously, a drug that has an effect on sympathetic control of the heart is going to be acting on the beta receptor, hence the name. And so I know that's where beta receptors are. But as far as I'm concerned, don't get bogged down with where alpha or beta receptors are. One of those types of receptors is found on the different organ systems, and they're activated by norepinephrine. Okay. All right. Is everybody with me so far? Any questions? Any confusions here? Okay, let me throw one last little thing in here, which is an asterisk right here. I'm putting that asterisk here, and you can't quite see it on the screen here, but there are a few exceptions to what I just said about the postganglionic sympathetic fiber. In some situations, the system doesn't have to quite conform to what's here. Now, there are 3 situations I'm talking about, and exactly what's going on in any of those 3 situations is not necessarily exactly what I'm saying, but that's okay. Probably what's happening in most of them is that instead of norepinephrine being released there, acetylcholine is released there. Okay. Now, with that much information, why is that a bad sounding thing? Or no, before we go to that question, to answer that question, to build up that question. Here, why are 2 different chemicals released onto the heart?
Different things.
They do different things. What does acetylcholine do to the heart?
Speed up. Speed up the heart rate.
No, it doesn't speed up the heart rate.
Blood pressure.
Lowers the heart rate. Parasympathetic system, rest. It lowers the heart rate to resting heart rate. Okay. And therefore, what does norepinephrine do?
Speed up.
Speeds up the heart rate. Okay. The heart knows what the heart knows, which is only what's chemicals it interacts with. It can't say, oh, that chemical is coming from the postganglionic fiber, which is coming from the ganglion fiber from the thoracic spinal cord. Okay. It can't see all the way back there to know where the signal is coming from. It just knows that's the signal. So on the heart, beta receptors or muscarinic receptors are activated by these chemicals. A chemical that activates the beta receptor speeds up the heart. A chemical that activates the muscarinic receptor slows it down. It's all the heart knows, it's all the heart needs to know. Okay. So if acetylcholine was released here instead of norepinephrine, what would the problem be?
The heart rate would slow down.
Right, the heart would know what to do because every signal it got would be the same. Okay. The exceptions that this asterisk represents are places where this kind of setup is not really going on. Now, I use the heart as an example here because it's kind of straightforward and exactly what I've said so far is true. If I changed this to a blood vessel, then things would be different because not all blood vessels do the exact same thing. I don't know if anybody's gotten this out of the reading or any of the presentations, but what does the parasympathetic system do to blood vessels? Anybody know? What'd you say? Dilates them. Okay. Allows more blood to flow through and blood pressure goes down. What would you expect the sympathetic system would do to blood vessels? The exact opposite. Constrict it, raises blood pressure. Is there a place that you can think of where you would not want blood vessels to constrict when the sympathetic system is active?
The heart.
That would be true. That's actually not where I was going for, but yeah, in the heart.
Brain.
In the brain. Other place I was not looking for, but also true. Yeah, I guess lungs too, but we're not to the one I'm looking for. When I talked about the fight or flight response, where do we want the blood to go? Skeletal muscles. We want to get oxygen to the skeletal muscles. Okay. So the first exception here is blood vessels in skeletal muscle. Now, the other things that people came up with, the heart, brain, and lungs, technically that's true, because you want more oxygen going to those organs also. But most of the blood vessels that do something weird in the fight or flight response are the ones going to the skeletal muscles, because there's just more of them than the ones going to the heart of the brain. Those want to dilate instead of constrict under sympathetic control, so that's where the blood goes. Oh, I'm sorry, it's going a little over. Give me 2 minutes to finish this thought, please. Mm-hmm. You'll be thankful for it. And so probably acetylcholine does that. So acetylcholine normally is a parasympathetic signal. And normally parasympathetic would cause vessel dilation. So for skeletal muscles, they get a sympathetic signal that acts like a parasympathetic signal. Might not be exactly right, but. Then what's another place where this kind of dual input is not found? That we've talked about.
Somatic reflex.
No, we're talking about the autonomic system.
Oh.
Where in the autonomic system is there not two inputs going to something to tell what to do?
Your skin.
Skin, which means specifically, remember,
Yeah.
skin piece here. What in the skin are we most concerned with? Yeah, the sweat glands. Now here, it might not be acetylcholine, but I don't really care. I haven't found a definitive source to say one way or the other. But sweat glands get a signal from the autonomic system. That signal tells them to produce sweat. If they don't get a signal, they don't produce sweat. It's not like the heart that goes up or down. It's not like they make more sweat or less sweat. They make sweat or they don't make sweat. So they only get one input, which is sympathetic. There's no parasympathetic control of sweat glands. Because you don't need to tell sweat glands to not sweat. They just don't sweat unless they get a signal telling them. Second thing. Now, the third one here is actually something that came up in the case that we did in lab. What was the unusual situation for sympathetic parasympathetic input that came up in the case? Anybody remember? Or let me put it this way. What was the most uncomfortable thing you had to listen to me talk about in the case study? Right. You probably did not really appreciate in the first week that you met me that I started talking about ejaculation, right? Okay. For the reproductive system, or since we're talking about the male reproductive system, we're really talking about the external genitalia. There's not a competing input there. You want the sympathetic system and the parasympathetic system both to do their jobs there together so that reproductive function does what it's supposed to. Now, the first two that I've written here are actually, oh, I changed the screen, but they're actually written on that table that I just had up there under that. The third one is not technically written in the book. There's weird reasons for why it worked out that way, but I'm telling it to you now. So, okay, I did not quite get to the end of everything I wanted to say in the autonomic system, so I'm going to pick that up a little bit on Tuesday to finish it off, which is mostly about the drugs that have an effect on the autonomic system. That is covered in the online content and in the book, but if you would like to wait till I physically say it in the room, you might want to hold off on submitting part 4 of the autonomic system. Okay. I don't remember if there's actually a question that depends on it, but don't submit it since you only get one attempt till after Tuesday. Okay. Do part 3, but otherwise, hold off on it. Thanks for waiting 5 minutes so I can give you that crucial information. And I'll see everybody on Monday or Tuesday, depending on who you are.